I guess you guys can see my screen, right?
Can you guys hear me again?
Yes.
Okay, groovy. Good, glad you got it working faster than I did. Yeah, I think it was just right-clicking on the new device and setting it for output. That would have probably fixed it.
Yeah.
Anyway, great.
All right. So if you can see my screen, we can start with the presentation. I mean, I want to keep this presentation more like the discussion we had yesterday. I'll cover some chunks, then we can cover any questions about those chunks of information, and then we can continue with the other chunks of information, so to say. That way, it's more like a discussion and not only me talking to myself for quite some time. Do you guys find that all right?
Yeah, perfect.
Yes. All right, cool. So yeah, the paper that Pi also shared, the Koha Code, the biological theory of memory. In this paper, I've written it a while ago. In this paper, I propose a biologically plausible way for how neural circuits essentially form and store memories. By memories, I mean like how they become pattern detectors. What's interesting is that it's heavily backed by actual biological evidence, like experimental evidence. One thing I would highly recommend, if you do not really care about the paper, it's not really written in a papery way. It could have been a blog post, honestly, but the patterns in the substrates subsection, that's actually the relevant part of the paper, because here all the highlighted parts cover very interesting experiments and citations. And yeah, the reference section is full of very interesting nuggets that might interest you. So the way I'm going to do this is actually, we're not going to look like the rest of the paper is cool, but the really relevant part is all covered in the introduction. The model part can actually be described using only illustrations, like from the slides here. Okay. So first, to cover the premise of the paper is that like the purpose of neural circuits is essentially to give rise to emergent algorithms. And by emergent algorithms, I mean cognitive abilities like forming memories, recalling memories, inductive reasoning, deduction, imagination, etc. And that trying to figure out how to reverse engineer those algorithms in a way. So the reason why I believe this is doable is because of the evolutionary forces that kind of give us some to reverse engineer the algorithms. So for example, if a neural circuitry has suddenly a mutation that enhances some cognitive ability, like observed patterns in that substrate, so in the neural circuitry, those are very interesting clues that can help us essentially figure out what's going on. So in most part, during the longest part of this presentation, I'm actually just going to cover those puzzle pieces, those clues, and then we can see how by actually combining those puzzle pieces, we can come up with a very simple and biologically plausible way for how to store, form memories, and how neurons essentially specialize to fire to specific inputs. All right, cool. So that being said, let's cover some interesting aspects of those clues that we can find. Before going on with the actual interesting parts like pyramidal neurons and interneurons, is there maybe anything anyone wants to ask or add or comment?
I'm curious what dates roughly these discoveries happened. Like one principal discovery in your paper is the capacity of a single dendritic spine to act as a convolutional filter on an incoming spike train.
That's actually my claim.
Oh,
Yeah.
is that actually, is there evidence for that, or is that a hypothesis?
Yeah, we will cover. So there's a lot, like countless papers covering those things. And even the father of neuroscience, Ramon Cajal or Cajal, I'm not sure how to pronounce it. Even he, when he observed dendritic spines for the first time, even he suggested that they might serve as memory storage units. So I'm certainly not the first to suggest that. Like even the father of neuroscience suggested that, but I'm suggesting that there's a hidden code within the dendritic spines that we have not identified yet. This essentially like learnable / modifiable temporal code that allows it to become this type of filter. For anyone confused with all this, Pi and I, we talked about the paper yesterday as well, so we will cover more about these details. But yeah, I will describe all the biology, like the mechanisms are there. Just that most neuroscientists did not really attribute those mechanisms to an algorithm, so to say. And I will cover those mechanisms in this presentation now.
I suppose the thing I was getting at is to get a rough sense of how the field of neuroscience is moving, like at what speed it's moving, were these main discoveries in the 60s and 70s and we're now just using them, or is there new innovation coming out from the domain of neuroscience? Is it
honestly,
at the moment?
Yeah, neuroscientists would tell you that things are developing really rapidly. But as a kind of outside observer, I find that most things are tend to get recycled. So there are a lot of things we knew even back in the 60s, 70s, 80s. And then in the 2000 something, like people would just rediscover something, attribute it to themselves, and get a lot of citations, you know. But when it comes, so we have done a lot of very valuable studies, of course, during the last 20 years as well. But things have not moved as fast as I would have liked. Especially a lot of scientists. So the field has, we noticed this in AI as well, where the field has, especially in academia, have people sub specialize in certain things, and then they have no clue what's happening in other areas of their field. And we've seen this, like, for example, theoretical neuroscientists very often create models that are not really that biologically plausible, but theoretical neuroscientists really don't like reading experimental papers that can disprove their ideas. And classical biologists / neuroscientists don't really like going into the algorithmic thinking part of things. And in a way, then what they do is they perform more classification than actual analysis, you know. So type of cognitive ability, I mean, we really have to be quite interdisciplinary. Like we have to read biology papers and we have to practice algorithmic thinking. Yeah.
Makes sense.
Yeah. All right. So I guess we all here are relatively good at algorithmic thinking, and that's why we will first start with basic biology here, because we have to have a better grasp at how the substrate that gives rise to cognition actually looks like before we come up with models to mimic it. And one very interesting thing, like let's start with the pyramidal cells, the neurons that we see up here, if you see my mouse. These are the most common type of excitatory neurons in mammalian cortical structures. So this is very interesting. It's also one of the biggest neurons actually in our brains. And essentially what excitatory means is that when these neurons fire, the other neurons that receive their signals tend to, if they fire, like they tend to be excited by the signals. The likelihood that they will fire will increase in a way. And this is quite interesting. And then another quite interesting fact about pyramidal neurons is that they're not only in human brains, like you find them in mammals and birds and fish and reptiles, like throughout the whole, like across the whole animal kingdom, we can find this type of neuron. So another type of neuron that gets very often ignored by textbooks is the interneuron, this neuron down here. Interneurons look quite different from pyramidal neurons, and they also have a very different function, as we will see during this presentation. So interneurons are also known as inhibitory neurons, which means that when their axon fires a spike train, so a series of action potentials, the neurons that receive that signal tend to shut off, like turn off. So they try to inhibit neurons whenever they send messages to other neurons, which is quite interesting. So if we can also go across the interesting facts here, and like once we have those facts, a lot of the things here make sense on a algorithmic level. But there are a lot of interesting, highly conserved patterns that we can find across nature when we start looking at pyramidal neurons and interneurons. One fact, for example, is that pyramidal neurons have dendrites. So for anyone not familiar with what a dendrite is, a dendrite are essentially these extensions that form synapses, i.e. connections to other neurons to receive input from them, okay? And the interesting thing is that pyramidal neurons have a very special type of dendritic setup, so to say, where like most non-technical people, like technical in the sense of neuroscience, would think that this extension here is the axon. At least I did that a couple of years ago. But actually, in the case of the pyramidal neuron, this extension is also known as the apical dendrite. So all pyramidal neurons have an apical dendrite, a very long dendrite that has a lot of smaller branches. And then they also have these, like at the soma, the cell body, they also have these basal dendrites. Okay. And then at the bottom here, an axon comes out and sends signals to other neurons. And dendrites are actually completely covered with some mushroomy looking things that are also known as dendritic spines. These were first observed by Ramon Cajal or Cajal. I'm not sure how it's pronounced. And at first, it was actually believed that it was just an artifact from the staining techniques used back then. But then later, people like scientists realized, oh, these are actual like components of the neuron. So the interesting next puzzle piece is that pyramidal neurons are completely full with these dendritic spines, like up to hundreds of thousands of dendritic spines per neuron. Whereas interneurons are essentially spineless. So it very rarely occurs that an interneuron has dendritic spines, which is, again, an interesting way neurons actually form synapses. So a synapse is actually the end part of an axon when it combines with a dendrite of another neuron, that small connection there. It's actually not a physical connection, but that small region there is known as a synapse. And what's very interesting is that the way synapses form are actually very different depending on what kind of neuronal configuration there is. So, for example, if an excitatory neuron, like the pyramidal cell, if its axon forms an synapse with another pyramidal neuron, it will always form the synapse on top of a dendritic spine, always. And so that's an interesting observation. So it will never form a connection to the dendritic shaft. It will always form a connection to the dendritic spine. Another interesting observation is that essentially every spine that we see does have a synapse. So it rarely occurs that the dendritic spine does not already have a synapse attached to it. Okay, very interesting fact. So, and the last interesting fact about the way these synapses are formed is that if we look at interneurons, the axons of interneurons actually never connect to dendritic spines, and that's very interesting because the interneurons will really actively try to avoid these spines and will instead try to connect directly to the dendritic shaft. So that's also a very interesting observation, and considering that it's very consistent across species, one would think that there is a reason for this kind of pattern. All right. There are also some other very interesting facts, which I would actually cover later on after describing how the actual algorithm works about the inside of dendritic spines. Pi and Wen, we didn't really discuss about this part yesterday at all. I will get to this in a moment, like after describing the algorithm. But before going into more like the neural backpropagation part and the actual model, does anyone have anything like a question or a comment or anything?
Quick question about that picture of various neuron types. Is that a complete classification as far as we know of what's in our brain? Is it just these two different types of neuron?
No, so that first great question. So we actually have a jungle of neurons. The number of neurons that we have is really large, and every layer of the brain can have different types of neurons, but essentially you can subcategorize neurons in different ways. And one categorization that matters a lot is categorizing them into excitatory neurons and inhibitory neurons. Now you can have various types of inhibitory neurons, and you can have various types of excitatory neurons, but it's like a major distinction. And another interesting thing is the pyramidal cell is the most expressed cell in our cortical structures, which is also the region where of the brain where a lot of the abstract thinking and memory actually happens. That's why I focus mainly on those cells.
Okay, I'm trying to get a sense of the composition of the brain. So is it like 40% pyramidal cells and 60% inhibitory cells, like by volume or by weight or what have you?
That's a good question. It's probably compartment specific. I wouldn't be surprised if different compartments have a different composition of the brain. I mean, it also depends. Like mass-wise, the pyramidal neuron is one of the biggest neurons in our brains, whereas interneurons are quite small. So mass-wise, pyramidal neurons would represent the probably I'm guessing the absolute majority.
Number-wise, would it be
Number-wise,
mostly inhibitory?
I actually don't know that. That's a very good question. That I haven't seen that in any paper actually. Like a paper discussed those ratios.
If it's useful or not.
Yeah, but I mean, there are though quite a lot of inhibitory neurons. So if we would look at the layer of pyramidal cells between the pyramidal cells, you would have a complete like mesh network of interneurons connecting all the pyramidal cells around them, essentially.
Almost as if the excitatory neurons are embedded in this
Yes.
soup of inhibitory tissue.
Yeah.
Yeah, gotcha.
All right, cool. So these were some of the major like biological puzzle pieces that we need to construct a biologically plausible learning algorithm. I will get to the dendritic spine in more detail after describing the model. But one other interesting phenomenon that we have to cover is a phenomenon known as neural backpropagation. So when I talk about neural backpropagation, I'm not really referring to backpropagation as we know the term in deep learning. Backpropagation, like in deep learning, does certainly not occur in biological systems. But there's this other very fascinating phenomenon known as neural backpropagation that is a phenomenon that is very often ignored by academia. In this, like, I guess that most people think that it's just an artifact, but there's very compelling evidence that this mechanism actually plays a very significant role in the brain, where whenever a neuron actually fires a series of action potentials across its axon, we will also notice a back propagating signal, like an echo, that will go through all the dendrites and across the apical dendrite and basically go through all the branches of the dendrite. This like echo-like signal. And why I'm saying that this is most likely not just some kind of unimportant artifact, but it actually plays a significant role is because if we look at the dendritic spine, after a backward propagating signal goes through the dendrites, we can notice something very interesting, that those dendritic spines that contributed in the action potential will have several hundred-fold increase in calcium ions within the dendritic spines. And that is a very fascinating observation, in my opinion. And what's even more interesting is other dendritic spines will not have such increased levels. Only those dendritic spines that actually contributed to the action potential will have increased levels of calcium ions. And not only like a little bit, we're talking like insane increase of calcium levels. Why this happens will become apparent when covering the actual learning algorithm. So yeah, now I'm going to go into the details of the actual algorithm. For that, we have to cover, we have to understand what these dendritic spines actually do. So in schools and in universities, students usually learn that the dendrite of a neuron essentially performs the integration of input signals, and then that integration happens. Well, some even think that it happens in the soma, but if we actually look where signal filtration or amplification occurs, we can actually see that it does not really happen at the soma of the cell. It doesn't really happen in the dendritic shafts of the cell. The majority of signal processing happens on a dendritic spine level. And I have included quite a lot of references of experimental data showing that that's actually the case. So essentially, what happens in a dendritic spine, and I guess I have to cover some of the facts about dendritic spines to explain the model, but what happens in the dendritic spine, we can imagine them as these mushroom-looking things, okay? And the dendritic spine also has a neck, this narrow space here, okay? A lot of studies show that this mushroom-like shape behaves like a biochemical compartment, okay? Meaning that it can separate the whatever kind of ions we see here from the shaft of the dendrite. And what's also very interesting is that the neck, actually the neck of the spine is the matrix of the dendritic spine is constructed with, it has a lot of actin, if it's pronounced actin. I don't know how it's pronounced actually as a word, but one very interesting property of this is that dendritic spines are very they can change their shape in seconds. So these spines can become more elongated or narrower or wider really rapidly, which is very interesting. And another very interesting fact, if it's pronounced actin, I don't know how it's pronounced actually as a well, first off, one important fact is that there are different types of spines. There's not only one single type of spine. We have references involved with all these processes. And here are the types of spines that I explained. So mushroom spines can last a whole lifetime. Thin spines have the ability to quickly appear and disappear. So it's believed, and one of the papers that I referenced here covers this in more detail, it's believed that thin spines have a more like a short-term memory role, and mushroom spines serve more for long-term memory purposes. And thin spines can actually become mushroom spines. So mushroom spines are like mature dendritic spines. And what's also very interesting is that almost every mushroom spine has a spine apparatus, that organelle that I mentioned, but thin spines do not have the spine apparatus.
So it's like 3 stages in the growth of a spine rather than 3 different types of spine.
Yeah, exactly. We should observe them more as stages, in my opinion. And like if we look at babies mainly have stubby spines. And if we look at adult brains, they mainly have mushroom spines. Yeah. So this is about the biology. Now that we covered this part, I will actually explain the concrete algorithm for how learning happens in neural circuits. Before jumping to that part of the presentation, does anyone want to ask or talk about anything?
Each spike in the same neuron have different pattern or activation, or all have the same, react to the same pattern of the same neuron?
Sorry, I some parts I couldn't hear well. Could you repeat the question?
Yes. The one neuron have, one pyramidal neuron have many mushroom or spike.
Yes.
But each of that spine have different pattern, or can learn different pattern, or all have the same pattern?
So my assumption is that they learn different patterns, because otherwise not that much information would actually be encoded in a neuron, and a lot of things that we see happen in neural circuits would not be really explainable. So I think it's way more likely that every dendritic spine has a different pattern.
Okay, yes, that got sense.
Nice. One thing that I did not really mention, it's more like an interesting fact. I will not really cover it in the model, but there's also another very interesting organelle known as the cisternal organelle. I don't know if it's pronounced like this. There are not that many papers that talking about it. You can probably count them with one or two hands. But this organelle is extremely similar to the spine apparatus of dendritic spines. So as a reminder, the refresher, the spine apparatus had the ability to quickly release and absorb calcium ions. And I'm arguing that the temporal code either somehow communicates with the spine apparatus or is somehow part of the spine apparatus. But it's actually the spine apparatus in like working together with the neck like matrix and the like synaptopodin protein and stuff like that that change the shape. Okay, and it was very interesting when I found out that the axon of pyramidal neurons also has an organelle that has the ability to quickly release and absorb calcium ions, because it didn't feel like we should observe it there. So if we look at this neuron, for example, all these things here are the dendrites, and this extension down here is the axon. And at the beginning of the extension, at the axon initial segment, you can find this organelle. And very interesting, there have been some interesting studies, these two references here, number 39 and 40, showed that if you would knock out the genes for synaptopodin in neurons, they would also not be able to form spine apparatuses in their dendritic spines, and they would also not have this axon, this organelle within the axon. And what they observed in mice was that mice without the spine apparatus and this thing that technically, in my opinion, is also a spinal apparatus, just for the axon, those mice were not able to form long-term memories. So there was a very clear link between the ability to learn long-term stuff and the spine apparatus, which is a very interesting observation, in my opinion. In my opinion, this organelle serves to figure out what temporal pattern to encode within the axon, but that's not really that important to understand the basic model of this paper. So okay, now let's cover the actual model. So now we're going to talk more about the algorithmic part, which is also very interesting.
Can I just check in so a neuron, it's listening for inputs over its dendrites, where each spine is listening to a different, listening for a different pattern. When it's got a pattern and it fires its action potential, is there some mechanism within that neuron that creates a specific encoding pattern, like a specific spike train that then gets sent out through the axon and then hits every postsynaptic neuron?
A great question.
And what chooses that pattern?
Yeah, so the current belief is that somehow all the signals received by dendritic spine somehow get integrated during like the apical dendrite, and then that pattern is fired out, which can certainly be the case. But personally, I believe that the temporal code of the output pattern, like the activation that the neuron has, is most likely influenced by that mysterious organelle within the axon that I just described. So my belief is that, and I don't have enough evidence for this belief actually, unlike all the facts about the dendritic spines, but I believe that every neuron has something like a signature, a temporal signature, and neurons essentially learn, just as in the case of embeddings, where embeddings start to learn to be closer in the embedding space to one another depending on co-occurrences. I think neurons learn to do a similar thing with their signatures, where every neuron has a signature, and then depending on which neurons co-activated with other neurons, their signature slightly changes over time.
It's almost like positional embedding in tokens for a transformer, that each token has a certain unique signature, which is its positional embedding, and then it has a kind of input-dependent signature, like the input modulates that signature somehow, maybe.
Exactly. Well, I wouldn't compare it exactly with the positional encoding of transformers, because it's a better example would be to compare it to the embedding layer of transformers, where there is already quite a lot of, like, every embedding that you have for a token for your vocabulary is depending on your training data set gets mapped in this highly dimensional space and becomes clustered and stuff like that. And the role of the positional encoding is actually just to tell the transformer then, hey, look, these happen nearby in time, whereas like the other tokens happened further back in time. But it's more or less like that. I believe that this mysterious organelle within the axon probably does something like that. Yeah. So I don't really cover much of this. So whenever because there are some other additional insights that I have not really included in this paper back then, but I think this organelle, together with the spine apparatus, are very crucial in memory formation. All right, cool. So
that's super interesting. I was completely unaware that there was this unit at the start of the axon.
Yeah, I guarantee you, even neuroscience students don't know this because it's not taught. I mean, you really have to go down a very deep rabbit hole to find a paper mentioning that. Yeah, so about the algorithmic part now. So now that we know all the puzzle pieces, so to say, about these highly conserved patterns across nature, we can start to think how we would design an algorithm, and that also takes into account all these things that we just covered. So, and this brings us to the Koha model. So it's lateral inversion. No, it's this section 2. Okay. The algorithm is actually quite straightforward. So imagine, so we have to take into account that the brain is an asynchronous system, unlike today's deep learning models. So there's no one single clock. Neurons essentially can receive inputs at different times. But one very interesting thing is that we discussed before how pyramidal neurons in a layer. First, pyramidal neurons in a layer usually have very approximately similar height within the layer, which is interesting. And they will be surrounded with interneurons. Okay, now what we can imagine is that, imagine that the neurons within this layer are receiving input from some previous cortical layer. Okay, now what actually happens is that always the apical tuft or how it's pronounced here receive the input, and the signals get processed by the dendritic spines. So if the input is very dissimilar to the code within the dendritic spines, it gets filtered out, and the signal is essentially dampened and slowed down. Whereas the neurons that have that are highly specialized to that input, meaning the temporal codes are very similar to the temporal code of the input, they will not dampen down the signal, and the signal can flow very rapidly across the apical dendrite. Okay, now you have to imagine this as an asynchronous, asynchronous process where every neuron is doing that, and the neuron whose signal first gets to the soma essentially fires first. And what happens here that is interesting, there is a configuration of interneurons and pyramidal neurons that is known as lateral inhibition. Lateral inhibition is briefly mentioned somewhere in the paper. It's just this paragraph, lateral inhibition, if you want to read a bit more about this. But essentially, it forms its configuration enables winner-take-all competitions, where this neuron was the first one who fired, and it will notify the interneurons, and the interneurons will turn off all the surrounding pyramidal neurons. And now comes the interesting part. Once this is done, the back-propagating signal of the winner notifies all the dendritic spines to become a little bit more similar to the input that they received. And this is what I hypothesize is happening when we see the super increased levels of calcium within the dendritic spines that take part within a competition. And essentially, this is all what the Koha model proposes. So it's proposing a competitive / Hebbian-like learning rule for neurons to specialize to become pattern detectors. Yeah, that's basically the idea of it. Any questions? Because this is the whole idea, but it makes only sense once we actually take into account all the biological evidence that we have. So this mechanism would explain why pyramidal neurons have this apical dendrite and interneurons don't have it. It would explain why pyramidal cells have dendritic spines, whereas interneurons do not have them. Because the pyramidal neurons are actually storing and forming memories, whereas the interneurons are more as relayers. Okay, and another very interesting fact is if we read this line here. Studies in the layer 2/3 of the rat neocortex have shown that pyramidal neurons possess about 10 times more local connections with fast spiking inhibitory neurons than with other pyramidal neurons, which would also make sense if in this model, because the signal across the pyramidal neurons moves slowly, whereas the signal of the interneurons moves really rapidly. Which in an asynchronous process with potential latencies is the kind of behavior you would want to see. So in a way, this model is very similar to how self-organizing maps essentially work. So if we search for self-organizing maps, they also use a competitive learning rule to have specialized units. And I argue that something similar happens in neural circuits in the neocortex. And this is basically the mechanism how it happens. Yeah, that's basically it. I think we can have a pre-discussion, whatever questions anyone has.
I have a question, quick question. This inhibition, it happens, like it actually inhibits or it shifts the timing that the spiking occurs into, like
it inhibits.
It inhibits, okay.
Yeah. So getting back to the comparisons we had with embedding layers, what we can imagine is if this so the claim that I made that neurons each have a signature when they fire, and they get closer. Like if we treat it like, instead of having a Euclidean space for the embeddings, let's say we have a Hamming space where like ones are the action potentials and zeros are the absence of action potentials. We can basically imagine that neurons that fire together will most likely, this is just a hypothesis, will most likely end up having similar signatures. So essentially their binary embedding would be close in that Hamming space to one another. And this also makes kind of sense. So if we have a cluster of neurons here and this neuron 1 here, and we have another cluster of neurons there and another neuron 1, and then they are connected to one another, that maybe the basal dendrites, because basal dendrites, these dendrites here have mainly local connections, so lateral connections, whereas the apical dendrite have more like across layers. But essentially, it could get the information, oh, look, the other neurons around me, these are the other winners. Let's modify my signature to become a little bit closer to them. I mean, for this last part, this is very hypothetical, but it's just a hunch that I have.
Do you think it would make most sense to model these things in layers or as a random graph?
Like
both.
I
know we have layers of cortical tissue.
So I think we would want to use both structures. So we would want to have layers of neurons, but each layer, in my opinion, should be more like a graph. So the lateral connections within the layer should be like structured as a graph, whereas between and then you can essentially stack different layers of graphs.
Also, what's the respective distance length of dendrites and axons? Is it that dendrites are capable of becoming extremely long and collecting a signal from a long distance away, but axons are quite short and you can only propagate the signal a small distance?
Almost right with all points. So axons can actually get quite long. Well, quite long in terms of like a cell level. I would have to look up the level of microns, but axons can reach distant regions actually as well. And the apical dendrite can also reach distant regions, whereas the basal dendrites, they mainly form local connections. So you can imagine like the axon can reach another layer and maybe like several other layers deeper as well, and the apical dendrite can also like reach previous layers and maybe even deeper layers, and the basal dendrites basically have this local information here.
When you say that the apical mushroom learn the pattern when the activation of the neuron, it's like a Hebbian learning.
Sorry, let me rephrase the question to make sure if I properly heard you. So are you asking if I'm claiming that
for the learning process if it is a Hebbian learning in the neuron?
Yes. So if you think about it, competitive learning is a form of Hebbian learning, just framed a little bit differently. So if we think about it, imagine this neuron won the competition, and through the back propagating signal across the dendrite, it informed its dendritic spines to update its code. What that means is that next time you receive an input from the neurons of the previous layer, it is more likely for this neuron to win the competition even faster than before, because it's now even more specialized. Meaning like the synapses can also become stronger and the dendritic spines can become like more mushroomy, so meaning more mature and stuff like that. So in a way, competitive learning in this sense is Hebbian learning.
Yes. What surprised me is that, or maybe I don't figure out that can be in the apical of one neuron. It can be different pattern that receive the spine. So different pattern can be learning to the same time from different neuron or another layer, and not just one pattern from.
Yes. So we should imagine people used to model neurons in a very simplistic fashion. We treated them as very basic perceptrons that just did some summation across some inputs, which we certainly know is not the case in reality. So it's more, a neuron is more similar to an entire neural network, artificial neural network, I mean, in complexity and in the ability to learn nonlinear like functions, so to say. Yeah, essentially.
Might it be reasonable to do a simplistic model where the neuron is puts out its dendrites as feelers, and the feelers pick up electrical charge, and the neuron accumulates this charge, And once the charge is above a critical threshold, it emits an action potential and then discharges itself through that action potential. And that over time, there's a leakage as well. So if it doesn't get any stimulation for a few seconds, it dissipates and kind of depolarizes itself.
I would say this is more how people used to think about how neurons receive and integrate information, but I think that the overwhelming evidence for the existence of temporal codes kind of refutes that.
What do we know about these codes? Like how big are they? How many spikes do they comprise, and what time period do they? How long is a complete train?
Well, that's the tricky thing. At least the studies I've read were observing a single neuron, but the code is actually like a single neuron actually receives many codes because it receives many inputs on different parts of the dendritic tree and like on the dendritic spines in that tree. Actually, I'm not sure if there are studies on that. Maybe there are some interesting studies.
Yes, that's what I'm wondering is what does a packet look like? If you think of it like a computer network sending data packets, then
I imagine it more like the burst thing that I mentioned about spike trains. One such burst could be treated as a packet in a way, like a Morse code of like the information is there.
Right. So I'm wondering how much is in a packet. Is it like 10 pulses over 100 milliseconds or 100 pulses over 1 millisecond? Or what's this variation?
But probably, I would be surprised if people haven't recorded. I just don't know the numbers, but I mean, we can search and try and find some paper that covers the millisecond aspect of
Yeah, I
it.
think that might be interesting to get a sense of the amount of information we're dealing with in a single transmission, and also the mechanism of the neuron in collating this incoming information and choosing what to spit out. That seems interesting as well. Like if we look at the signals coming out of a particular neuron, are they similar to one another? Do they have like a unique fingerprint characteristic, or are they entirely different depending on different inputs? How is that output signal composited?
Yeah, and keep in mind, it's always very tricky to make those studies without corrupting the system, because if you force a neuron to fire, you will actually overwrite its weights, its code, like the temporal codes. So ideally, if someone would design some experimental setup, you would want to make some like more old school experiments where, for example, in the book that I shared today in the Discord server, brain vision, no, how was it called? Eyes, brain, and vision, or something like that.
Yes.
Yeah. If you, I forgot which chapter, but there were some experiments on, I think, cats where they wouldn't really force activations on the neurons, but they would show it different inputs, like through its eyes,
Yes.
and then they would observe which neurons are firing. And that's like the type of experiments we would want to do, because we would not want to interfere in the activations. Yeah, but yeah, that's more
Yes,
something
you want to
for
be witnessing what goes in and what goes out without forcing it.
Yeah,
which is going to be very difficult.
and I didn't really cover, like in this paper, I also briefly talk about invariant representation learning, receptive fields and invariances. I didn't really cover it because I actually don't think that is that relevant to the paper. If I would have written it now, I would probably have left it. But it's very important to keep it in mind. So neurons, the artificial neurons in deep learning models are actually very different from how neurons in biological systems behave. For example, like the brain has some cells, we call them simple cells. We divide them into simple cells and complex cells, where simple cells are essentially like in the case of in convolutional neural networks, there are simple pattern detectors. So if this here is the receptive field, whenever it sees, for example, an edge or a line in the center of the receptive field, let's say, it fires, it throws that like burst of spikes. Whereas if the pattern is somewhere else in the receptive field, it will either fire very little or not at all if, for example, you shift in this case, the line or something like this. So this kind of behavior we can mimic pretty well in today's artificial neural network models. But there's also another type, and the majority of cells in our visual system are actually made out of this type of cell, which we call complex cells. And complex cells have the ability to learn very complex representations that have very often some invariant capabilities. For example, there are neurons in our brain that will fire whenever a line is seen in the receptive field, regardless of the position. So this is also known like as translational invariance. Like in convolutional neural networks, we have translational equivariance. And then we have cells that will fire regardless of the size of the pattern that it sees. So we have shape or size invariance. Then there are also other types of representations where a neuron will fire whenever it sees a pattern move to one side of the receptive field, but it will not fire if the same pattern is moving to another direction within the receptive field. So here we have also some temporal aspects of specialization. And then, of course, we also have even more complex stuff like viewpoint invariance or luminescent invariance, where a neuron will fire whenever you see Bart Simpson, but it will not fire when you see Homer Simpson, for example. And these, I think we really have to try and figure out how to design systems that can learn in an unsupervised fashion as many invariant representations as possible, because these representations are quite rich and can help us design models that require less data to train, in my opinion.
But this behavior, this complex behavior, isn't it just the, sorry for just saying, but isn't it just the position, because of the position of the given neuron, like all the previous neurons are pre-processing the data, and that's causing the given neuron to fire at Bart Simpson. It's not like the full complex computation happening inside the one neuron, right?
Yeah, so definitely, it's not just like the result of one neuron, but the result of a lot of neurons sending specific signals to one neuron that just specializes. In this case, for Bart Simpson. But I haven't seen anyone yet come up with a generalized approach / algorithm for how to achieve that. Because if we could mimic that behavior, that would be quite in without having to hard code stuff. So that would be quite amazing. Because we have learned
reward invariance.
Yeah, so we know how to mimic some of these invariances by hard coding some rules. For example, convolutional neural networks have certain inductive biases that enable it to have this translational equivariance, which is cool. And sure, you can also modify your data set so that the neural network learns to become, let's say, rotation invariant or something like that. But all those feel to me very similar to how computer vision worked before 2012, where we really try to manually make filters and pattern detectors and stuff like that. And then convolutional neural networks came and it made all that kind of irrelevant, And we need a transition like that, in my opinion, for the invariant representation part as well. And And one thing that we also briefly discussed in yesterday's call is that with Boyan, I'm very confident, like certain, that the only way how to learn these invariant representations is by making use of the temporal information of your environment. So, for example, if you have a cup and you look at that cup and you can move your head and look at it from different perspectives, your brain might learn that, oh, okay, all these different angles and perspectives are belonging to the same thing / concept. And I believe we essentially make use of that temporal information to learn these invariances in some way.
Just
If you
one more.
put in Hubel
Sorry.
Weasel cat experiment into YouTube, you can see a simple 3-minute video of the cat experiments they did, where they've got an electrode wired connected to a neuron in the cat, and they're showing it a line of light that's crossing the screen vertically and diagonally and horizontally. And when it crosses diagonally from left to right, this detector, it's like a Geiger counter. It goes click, 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 click,
Yeah.
etc. So I'm wondering if there's something special about the delay between the spikes, or whether it's just a basic rate. It's like a way of sending information, sending a certain amount of electricity, and it might take 12 spikes to send that amount of electricity, but then it might not actually be important, the actual delays within that spike train, or it might.
Well,
I don't know.
I mean, I know that maybe I even referenced some of those papers in the Koha Code paper, but I know that there have been studies showing that actually the distance between those clicks matters. So even if you have the same rate, some neuron might not fire.
Oh, that's really interesting.
So it seems that at least for some regions in the brain, there seems to really exist a temporal code, but I would not. I don't want to have a very set in stone opinion about this. So it might also be the case that we have different encoding schemes in our brain. Like maybe some compartment benefits evolutionarily more from having a temporal code or information theoretically or whatever, and some other part maybe can make use for whatever reason also from the rate coding hypothesis.
So there's one thing that concerns me a little in all of this, is that I don't see a ready biological mechanism within the dendritic spine that would be capable of doing a convolutional filter for a specific sequence. So what I wonder is, maybe it might be multiple spines acting together does a complete pattern recognition of a complete sequence, and maybe the individual spines catch something similar, like if each was a frequency detector, and then the composite of many of these would catch a certain frequency signature. I wonder if it might be something simpler that happens within a particular spine. Is there
Can we say that, like,
there's
the
a filter?
can we say that the distance between the dendrites has something to do with this coding that Pi is saying, or like the distance doesn't matter? Because what I know is that there can be multiple connections between like the same neurons, and I was just wondering, like, does the distance, like the total distance that they have, make a difference into this temporal coding, or is that not really relevant?
You mean the distance between one sending and one receiving neuron, or you mean the distance between the spikes within a spike train?
I mean, the actual physical distance between like the synapses between two neurons. Like two neurons can have more than one synapse, right? And they have different lengths, physical lengths. Does that play a role into the temporal coding, or is that not relevant because the distance is not like too difference between those two synapses?
So I would divide this question into two answers. When it comes to the signal of ascending neuron and the neuron, let's say between two layers, I would assume that as long as the neurons within a layer have an approximately similar distance to the neurons in the other layer, it doesn't really matter. But there is some interesting evidence. I don't remember where I read this though, where if we look at the dendritic tree here, so this is how a dendrite looks at close-up. So it's full with these dendritic spines, and each dendritic spine, you can imagine each dendritic spine being a synapse. So it receives some input from some neuron somewhere. And what has been interesting, and if I recall it correctly, is that there is some type of voting happening here as well within the shaft. So if this spine is receiving some code that overlaps really nicely to its internal code, but it's not observing at the spine neck level that any other signal is coming down the shaft, then it will kind of dampen the signal. So in that case, co-activations matter a lot. So if only a very tiny subset of a pattern is used, it might not activate the neuron. You need one part of the pattern might reach this dendritic spine here, another might reach this dendritic spine there, and whatnot. And if the neuron actually fires, matters a lot how complete that pattern is. If this kind of answers your answer.
Yeah, so like on figure 9, it's depicted that the like temporal pattern, like it's inside dendritic spine, like the internal code. I don't know enough to picture how that can be like physically inside the dendritic spine. So my hypothesis was that the temporal coding is not only in one dendritic spine, but across like a group of dendritic spines. And the physical distance would act as the temporal pattern, how this temporal pattern would evolve.
Yeah. So that hypothesis, I think, could be plausible as long as there's somehow a mechanism within the dendritic shaft to activate the spine apparatus within the neck. So if there is some compelling evidence for that, your hypothesis can be like highly probable. So in this sorry, one thing first. So in this hypothesis, I'm assuming I'm assuming that it's simpler to assume that the spine apparatus somehow has the ability to detect spike trains, and then the spine apparatus can decide if it wants to release calcium ions or absorb calcium ions. But as long as the spine apparatus, we know that the spine apparatus has some extensions that go within the dendritic shaft. So it could also be the case, as you're saying, that somehow it receives the information within the shaft, and that shaft information tells the spine to perform the absorption/release. But I haven't seen a lot of evidence for that, so I thought that this design might be more plausible.
So I've got a hypothesis here of how it could be made to work, that an individual dendritic spine, it would first activate on the first impulse of the spike train, and then it's like a delay filter, and the width of the neck, the tightness of the neck, controls the delay that it's looking for. And if it gets a second if it gets a pulse after that delay, then it fires. So now if your train comprises 10 spikes, then 9 dendrites could be used to get the delays of each of those following 9 spikes from the first spike. Each of those dendrites would be activated on the first spike. It would listen for a certain delay, and if it gets a spike after that delay delta, it fires.
Could
So if you get
be
that exact timed spike train, then all 10 of these delay filters would fire.
could be the case. I would also not rule out the hypothesis that technohybrid proposed. So even though in this paper I chose one possible explanation, I'm very open to other possibilities. But as long as those possibilities can be explained with the biological evidence that we're seeing.
So how many connections are we looking at between, so say neuron A connects to neuron B being the postsynaptic neuron. How many places might neuron A connect to neuron B? Might it be like 10 synapses, 100 synapses, 1,000 synapses?
I've read I think in the book Dendritic Spines, but I'm not sure. This book, by the way, I highly recommend Rafael Justus' book to anyone interested in this topic, even his papers. Rafael Juste was also the first guy that covered that the backpropagating signal does have an effect on the dendritic spines. Maybe someone else did cover that before Rafael Juste, but he really wrote some nice papers about that. But getting back to your question, I think in his book, in this one, if I'm not mistaken, it's described that neurons, and this also makes information theoretic sense, the neurons try to connect to as many possible other neurons as possible, which means you don't want to have several connections to the same neuron. So somewhere I read that on average, like every neuron has only one connection to some other neuron. But don't quote me on this. I might be also wrong.
Because if that's the case, that would rule out the theory that I proposed. But
which one?
My suggestion was that maybe neuron A connects to neuron B at say 20 different on 20 different spines, and each of those spines is looking for a particular delay. And so if you get the right sequence of delays, all those 20 spines fire.
That seems highly unlikely to me. Like how I imagine it is more that like you have a distributed pattern of neurons activating, and every neuron tries to capture as many of those firing patterns as possible. By capturing, I mean form connections to those neurons and try to specialize to either dampen or like amplify those signals. You mean what?
Work to push into the dendritic spine to get it to act as a full convolutional filter. It would have to somehow store temporal information for the spike train that it's trying to listen for. And I don't know, is there any evidence in the biology that has sufficient structure?
I haven't seen. So this is actually the main hypothesis of the paper. I'm claiming 1, there is a code, and 2, it's that code is either somehow tightly linked with the dendritic spine internally or somehow externally and communicates with the with the spine apparatus, I wanted to say. Because the spine apparatus is essentially what's deciding if the spine neck is expanding or like becoming more narrow or elongated or whatnot. So I haven't seen anyone else contemplate it this way, because most people don't even know about the spine apparatus, honestly. But considering that the dendritic spines can really rapidly change shape, and they change shape differently based on the input that they receive, to me seems like very compelling evidence that the spines know somehow when to have preferential treating towards a spike train, or like when they receive it.
I like the idea that, say, we're puzzling on a math problem and we're just staring at it and nothing's coming to us. And our little mushroom dendritic spines are elongating and contracting until at some point they get a lock on the problem and they spot the pattern and boom, a signal fires. And a back propagating signal says, yes, you got it, just fix those positions. Those were useful positions that you just found. So a very tiny subset of these spines lock their positions because they got something.
Yeah, and there's this one quote in the paper that I have to find it. I really like that quote. Where was it? Yes, I think it was this. Yes, individual nerve cells were formerly thought to be unreliable, idiosyncratic, and incapable of performing complex tasks without acting in concert, and thus overcoming their individual errors. This was quite wrong, and we now realize their apparently erratic behavior was caused by our ignorance, not the neurons' incompetence. This quote by I think a Hungarian scientist. I hope I haven't missed, like, number 48. This dude perfectly sums up neuroscience, the how things moved in neuroscience, because until really, like, well, even in the far past, there was more than enough evidence that should have helped us overcome our ignorance. But the dominant narrative, even today, I would argue, is that neurons are way simpler than they actually are. I think that it would be a way healthier approach for people dealing, like, in neuroscience, but also in AI, who are interested in neuroscience, to imagine neurons more like today's artificial neural networks rather than today's simple units, so to say.
I have maybe a dumb question. So, like, you were talking about simple versus complex neurons in the visual cortex. Is this true for the rest of the brain? And is, like, are there any specific, like, areas where you have more complex neurons with more invariances, like weirder invariances?
Yeah, I think so. I wouldn't be surprised if there is no morphological difference between simple and complex cells. It's my guess is that so you definitely have complex cells in, for example, in the auditory system as well, because there are some, like, you will know Pi's voice regardless of if he has, let's say, a cold and his voice is slightly different or something like that. You will associate it with Pi, and that is a type of invariance in this case. My assumption is that probably the visual system has the most complex cells, but because of simple information theoretic reasons. We in the case of vision, we're actually receiving the 3D information, and we have also the temporal aspect to that. And you can simply extract way more types of invariances from that type of input. And in the case of the auditory system, you have fewer dimensions, so to say. But I don't think that there's something inherently different between the neurons of those systems. That would also explain why it's very simple for example, if we look at the brains of, for example, blind people, the parts of the brain that would deal with a visual system really rapidly readjust and are used for the auditory system. So makes me think that it's the same type of algorithm that processes these things.
Okay.
So we're rewarding the system to find invariances of a different nature, because if two invariances are roughly the same, the competition will rule out one or the other. and we're also rewarding it to find the most efficient path to finding that invariance. Because if two paths are found, then competition rules out the slower path.
Yes, and that's where the thing with the energy constraints that Brian mentioned yesterday would come in. Competitive learning really is helpful in trying to perform the most energy efficient computations for some certain tasks then. Because in the real world, I mean, energy, how much energy you waste per cognition matters.
Do you think you could reach out to a neuroscience lab and tell them the experiment that they need to design, where you hook up an electrode to the pre-synapse and to the post-synapse and monitor the ability of a dendritic spine to capture a spike train? I think that's experimentally verifiable.
Yeah, back in 21, I actually tried to reach out to some neuroscientists. I quickly realized that most neuroscientists don't have the proper training in algorithmic thinking to appreciate the suggestion. And two, there's a very strong authority bias in academia, especially in neuroscience. And for them, I'm a nobody. So it would have to be a figure of authority for most neuroscientists, I would argue. And I just wasn't lucky. I actually got attacked by a lot of people, even though I tried to really cover if you look at the references, there's so many references I'm trying to include here, and each one is about biological experiments, and I still got attacked like crazy.
I'm sure you trod on a lot of feet, a lot of vested interests.
Yeah, because I mean, imagine someone spending 7 years in a PhD about something that turns then out to be wrong. You know, it becomes then an ego problem.
I wonder if companies like Neuralink or the other companies that are working on brain-computer interfaces might be more open to this kind of experimentation.
I would
to start, they've got money, and then they've got an interest to make something that works rather than to protect some theory.
Yeah, I would expect the industry to be more open to new ideas. I just haven't really tried to contact anyone from the industry. I don't know anyone in the neuroscience field, actually, because I'm not a neuroscientist, you know.
Yeah.
I
have a feeling that would be a very informative experiment to actually figure out the amount of computation one particular dendritic spine does. Can you, if you had the resources, do you think you'd be able to set up the experiment
myself?
Can you see a path through that?
I definitely not. Performing experiments in wet labs, because I worked 1 year long in a wet lab, but as a bioinformatician. The engineers, we like to underestimate how difficult that part is. It takes quite a lot of experience. I don't have that experience.
But you reckon it could be done for someone that's in the field?
Oh yeah, definitely. Someone who's in the field and who has worked with dendritic spines before could certainly do something like that.
That seems like a really good avenue of inquiry, even if it's frustrating and you have to deal with a lot of gnarly people. If you could finally get through to someone that's director of operations in a lab that has the equipment and expertise to do that experiment and can wire it up.
Yeah.
And there would be some interest for them as well, that they would be prominent in a groundbreaking publication.
Yeah, certainly. I mean, right now, I think I'm gonna invest more time into the Koha Code hypothesis paper once, only after like Diane and I, we're working on various AI related things right now, and I figured out, like, in my opinion, it would be easier to convince people in the neuroscience fields once we have impressive stuff in AI as well, because most people unfortunately have a monkey see monkey do approach. Like, there's, for example, no evidence whatsoever for backpropagation in the brain, but just because backpropagation is so successful, there are quite a lot of neuroscientists who actually think that it's possible, even though there's absolutely no biological evidence for that. So I think a similar approach might be helpful, where if you show that, look, what you can achieve these things with competitive learning, people maybe are more open towards the ideas proposed in the Koha Code paper.
Do you think you'd be able to make a simulation of a dendritic spine and arrive at a strong hypothesis by running the simulation? Like, do we have it under the microscope well enough that you might be able to figure out how it might be storing information?
When you say simulation, you mean like trying to replicate all the molecular structures we know, or
something like that? Just something good enough to suggest how much information it might be able to store, whether it is capable of applying this convolutional filter to an incoming spectrum.
I don't think that we have enough knowledge about that. So there are too many unknown unknowns. So imagine, for someone who does not even know the complexity of genetics, and inheritance and DNA, when they look at the chromosome, it's just structure. So you might simulate that structure, but that doesn't tell you anything about the mechanisms that happen within the chromosome,
Yeah.
Yeah. But what's interesting, mushroom spines, I've read the studies somewhere that showed that mushroom spines do have ribosomes. So they have the ability to synthesize proteins on a spine level, which is interesting because that means that different spines can have different expressions, protein expression. So that's quite interesting.
So I have a question still. Regarding like figure 9, and the whole learning process of neuron becoming specialized to a signal. Up until now, I wasn't aware of a backpropagating signal. What do you think would regulate the intensity of like this specific neuron learning or becoming more specialized to detect a signal?
I'm not even sure if there is such a thing as intensity of learning in neuroscience. I mean, there might be, but I wouldn't assume that that's the case. Maybe it's like a constant thing. Who knows? Like, okay, you won the competition, update your dendritic spines by this rate,
How can we incorporate the observation that a brain, even in an inactive state, tends to have roughly the same amount of firing as a brain doing a task? Is that even a correct statement?
Well, it depends how you define inactive brain. Like, you mean if we're just sitting around, or do you mean a coma?
Doing nothing with your eyes closed, not thinking thoughts, then there's a certain firing rate in your brain.
I would argue that you are thinking thoughts, you're just not aware of them. The conscious part of ourselves is a very small fraction of the actual processes that happen in our brain. one thing I discussed with like Dian maybe a month or 2 months ago was the split brain experiment. Dian, do you remember which YouTube channel made a video about the split brain experiment that we watched together?
I think it was Veritasium. I'm not really sure. Probably it was that one.
Yeah. So maybe I can share it afterwards. But there are patients. So we have two hemispheres in the brain, and they are connected. And there are some cases where for medical reasons you had to cut off the connection between the two hemispheres. And what's fascinating is that language is only in one side of the hemisphere, so the other side does not have access to language-related cognition. And the experiment really is interesting, where you might pick up a shirt, and your left hemisphere likes that shirt, but then your right hand just comes up and throws it away because the right hemisphere doesn't want that.
Okay.
Or for example, the right hand does something, and then the other side, where the language center is, has no idea why it did that, because there's no connection anymore, but it starts to rationalize that action. So anyone interested, split brain experiment, it's fascinating, and that kind of shows us that we're less of a single entity as we think. We're a collection of different parts, trying like a consensus, figuring out how to behave like a unit, you know. So even if we remain idle, even if your conscious part might not be thinking something, I would be very certain that the rest is still processing stuff.
There's interesting research in transcranial magnetic stimulation as well, that if you apply an electromagnet to one side of the brain, you effectively deactivate it.
Yeah.
And then you can, so they got people to draw a picture of a horse, and it was terrible, like a 2-year-old. And then they apply this stimulation for 20 minutes and do it again. And the image is much more like an artist would render it.
I haven't read much about this topic, but I know that people can even induce flow states in people. So especially, I wouldn't be surprised if the US military has invested a lot of money in this type of research, because they have been researched. As long as the soldier is in the flow state, their performance is like absolutely like they're a one-man army. Whereas when they're not in the flow state, they're regular humans, you know. I have no idea how that works. Or there have been studies where you can turn people into atheists with that technique somehow, temporarily. Or maybe was it reverse, believe in the presence of something in the room. So very, very fucked up stuff. I have no idea, but it's fascinating. Yeah. So I think we can pause the recording, because I'm also a bit hungry now.
Thank you very much, Chainless Coder, for that presentation.
Thank you.
Yeah.
Brilliant.
Thank you for joining this paper discussion.
Thank you.
And yeah, we'll